Hello, everyone. Good morning, good afternoon, and good evening to you, wherever you're joining us from today. Thank you for being here. I'm Shefali Lathwal, and today I'll be talking to you about, you've guessed it, ontologies, but more specifically about how we use ontologies at Elucidata and what the use of ontologies enables us to do. So I'll be focusing a lot more on applications of ontologies today. So let's dive straight in. Now, before getting to the ontologies part, I want to set the context that we will discuss today. Gabriel, in her presentation, talked about electronic lab notebooks. At Elucidata, we primarily work with the life sciences R&D data. And life sciences R&D domain in general is extremely heavily data-driven. Electronic lab notebooks are part of it as well, and there are several other types of data that feed into life sciences R&D too. But at Elucidata, we primarily work with biomolecular data, which includes various types of omics data, such as single-cell transcriptomics to bulk RNA sequencing, proteomics, metabolomics, genomics, flow cytometry, drug screening, and several types of bioassays as well. And a large amount of these data today exist in public domain, but a lot more actually are held within organizations as well. And every year, several trillion gigabytes, which is a number that's very, very hard to imagine. But that's the scale at which data are being generated every year for these different types of R&D projects. And while so much of these data already exist and are being continually generated, there are actually several barriers that prevent researchers and teams from using these data to their full potential. First of all, even if data exists, you need to be able to have access to the data, which means that the data licenses need to be very clear. You should have permissions to use the data, or you should be able to apply for those permissions. Secondly, you don't just need to have access to the data, you also need to be able to find the data. And this is a particularly challenging problem that we are going to dive into a little bit more today, because a lot of data that exist are actually siloed across many different sources. And that's true even within a single organization, where data can be scattered between different teams and between different data management systems, between different file management systems, and so on. And the same is true for publicly available data as well, which are scattered across different repositories. And it's very hard to keep up with the new data that are emerging, either present in the public domain or even within one's own organization. And thirdly, if you're very lucky and you're able to find the data that you need for your research, you should also be equipped to actually work with that data, which means that you should be able to use the metadata to understand what the data contain, to evaluate the quality of the data and its suitability for your needs, as well as to you will need to make sure that it's in a format that you can work with, that your machines can work with, and that you have the computational resources to be able to handle that data. And only if all of these conditions are met, can you take existing data and reuse it, and reuse them to generate new insights. And a lot of these hurdles to data usability are actually encapsulated in the acronym FAIR that Gabriel also talked about in her presentation. And a big goal at Elucidata is to make the existing data more fair, which means more findable, accessible, interoperable, and reusable. Now, while there are many reasons that data are not usable, as we just discussed, but one of the main ones that I want to focus on today is lack of metadata standardization. And when I refer to metadata, I'm primarily referring to information about an experiment. For example, what is being measured, whether they're proteins, genes, metabolites, and so on, what biological system is being studied. For example, is the experiment being done on humans, on mice, on yeast, where the samples are coming from, so the tissues and other information about the experiment, as well as the metadata of the samples contained within the experiment, and how they were treated. For example, was a specific drug given, at what time point the data were collected, and so on, as well as information about the molecules or any other entities that we are being measured. So the example on your screen, you can see that it's a gene expression data set. So we need to know the metadata about the genes and what the expression of each particular gene was. So if any of these three types of metadata, the experimental metadata, sample metadata, or the feature or molecular metadata are not standardized, which they very often are not, as we'll see in just a minute, it becomes very, very difficult to find the data that you actually need, and then to use these metadata to evaluate the quality as well as the suitability of data for your needs. Now, when we say that the metadata are not standardized, here are a few examples of what that looks like in practice. So in day-to-day practice, there are many different ways of referring to one single concept. For example, hepatocellular carcinoma, which is a disease, is referred to by many, many other names like liver cancer, liver neoplasms, hepatic neoplasms. It's not just different terms, but even abbreviations like HCC, and all of these are perfectly acceptable in day-to-day conversations, and we'll find examples of data sets studying this disease annotated with all of these different terms. Similarly, there are many different ways to write a single word. For example, the word non-smoker can be written with a dash, without a dash, with a space, without a space, and so on. So some heterogeneity is because there are different accepted terms to refer to the same concept. But a lot of other heterogeneity is also because of lack of clarity on what a metadata field should contain. For example, a field like smoking status can have values like yes, no, values that contain actual meaning like smoker or non-smoker, and even numbers like 0 or 1. There are also data entry mistakes and misspellings that lead to heterogeneity in metadata, as you can see in the case of gender here. And the result of all of these different conventions and this lack of standardization is that a very small portion of available metadata are actually standardized. And on top of that, these metadata fields are often not filled and are missing. So given that this is the state of metadata today, where metadata when present are not standardized, and often they're actually absent in many cases, the question we asked at Elucidata is, what can we do to make these data more usable? And the approach that we chose was to use natural language processing to fill in these missing annotations wherever they were missing, and then to use ontologies to standardize the metadata terms. So what we do is that we use the information that is available and present in a data set, and we use natural language processing to identify key concepts, key pieces of information that are important to understand what the data contain, such as disease, as you can see right now. And then we standardize these concepts using ontologies. So in this example that we are showing, we identified different ways of referring to COVID-19 and then mapped these different terms, different conventions to a standard disease ontology term, which in this particular case is from medical subject heading or mesh ontology that we use for diseases. And we've done this sort of metadata harmonization, not just for disease, but for several different metadata fields. And we use different ontologies for different types of metadata. And I'm showing a few that we use currently. We use mesh for diseases. We use NCBI taxonomy, Cellosaurus, cell ontology, Brenda tissue ontology, and chemical entities of biological interest ontology, and several others as well. So, so far, we have used this approach of using natural language processing combined with ontologies to curate 14 different types of metadata, so 14 different types of information across 26 different types of data. And by types of data, I mean different types of omics and bioassays data that I discussed earlier. And we've done this standardization for more than 1.5 million data sets. And in this process of curating these large numbers of data, what we've done is we've built tools and technology that can now enable us to do this curation for any new data as well. And at the end of this process, what we achieve is that we go from the state of missing and heterogeneous metadata at source to a much more usable data resource that can power biological discovery. And we will look at a few examples of what these applications look like. Now, before I go to specific use cases of what curation of metadata through ontologies enables us to do, I would like to take a minute to talk about why we use ontologies for curating metadata. So Gabrielle also mentioned and defined ontologies in her presentation. And there are several different definitions of ontologies that exist, but what I really like to do is focus on what are the key features that ontologies have that make them very attractive for standardizing and curating metadata. And one of the big ones is that ontologies actually eliminate subjectivity because they have unique identifiers defined for each unique concept. So earlier we saw that there are many different ways to refer to a single concept like a disease, liver cancer, which can be written in many different ways. But a disease ontology will actually have one identifier for this disease. Which is very appealing to people working with data because you always want to standardize and eliminate that subjectivity. A second big reason that we use ontologies is that they have predefined relationships between different concepts. And these relationships are extremely important because they represent current knowledge in a domain, and they also evolve and change as knowledge in a field grows and changes. So for example, the fact that liver cancer is a type of liver disease, as well as a type of cancer, will actually be embedded in the disease ontology. And if tomorrow there is a new subtype of liver diseases that is discovered, ontologies can be updated and that new knowledge can actually be embedded within the disease ontology. A third really critical piece for us is that ontologies are both machine readable and human understandable. So machine readable is really critical because we are working with data at very high volumes, at very large scales, and because ontologies are structured in a very specific way so machines can read them, it means that we can integrate these ontologies into our data pipelines, which is how we currently operate. But equally important is that the concepts in many ontologies are actually also designed to be understandable to humans. And that's really important because when we apply these ontologies to specific types of metadata, we can actually look at these concepts and we can evaluate whether a specific ontological term makes sense in the context that we are using that ontology. So from here, let's spend the remaining time talking about what metadata curation through ontologies actually enables us to achieve at the end of the day, and how do we apply ontologies in practice at Elucidata. So I will talk about 5 different use cases today. The first use case is that because ontologies have these predefined relationships built within them, they enable us to look at data at several different scales. And what that means is we can look at broad categories as well as very specific terms. For example, here I'm showing a snippet of the disease ontology that we use and the broad disease term cardiovascular diseases. And as you can see here, the ontology actually contains these predefined relationships that not only capture this broad disease, cardiovascular diseases, but also more specific subtypes of cardiovascular diseases. And once we've used this disease ontology to then curate our metadata, we can look at a high-level view of the data, which means that we can look at all the data that we have related to all types of cardiovascular diseases, which would include all of the subtypes, as well as a more granular view of the data, where we can specifically look at only coronary diseases or myocardial infarctions, depending on our use case. And this is really critical because this is just one example. For something like disease, there are thousands of these disease terms that exist, and the fact that ontologies capture relationships between those disease terms make them way more usable for researchers as well as for computational systems. Now, a second really big advantage that ontologies provide us is that they help us make the search experience on our platform a lot better. So the platform that we have, the data that I just spoke about that we curate using ontologies is put on a data platform where all of this data can actually be searched. So if we search for a term such as inflammatory bowel disease on a platform that actually does not use ontologies, as you can see on the left of this slide, then what will happen is that we will only get results based on direct text matches. And as we saw earlier, this will miss a lot of data because there are several different ways to refer to the same disease. Now, some data sets may actually contain the term inflammatory bowel disease. Others may only contain the abbreviation IBD, for example. And therefore, data sets that are not annotated with this exact term will not be part of search results. On the other hand, if we have used natural language processing to identify all the instances and variations of this term, inflammatory bowel disease, and then we've used ontologies to tag the data, then we get results that contain both inflammatory bowel disease and IBD, as well as all the other variations of this particular term. And not only this, what we can also do is we can use the relationships that are between disease terms that are embedded in a disease ontology to include more specific subtypes of inflammatory bowel diseases, such as ulcerative colitis and Crohn's disease, which otherwise would be completely missed. And this is how we actually power searching through the data that we have on our platform. Now, a third and again, really powerful advantage of using ontologies to standardize the same concept across data that were generated independently and are coming from completely distinct sources is that they allow us to examine and compare the information that is contained in these sources. So here I'm showing 11 different data sources where we have used mesh disease ontology to standardize the disease metadata. And because we are now using the same vocabulary for disease in each of these sources, which otherwise were generated completely independently and don't talk to each other, we can now put them all together and see which diseases are overrepresented or underrepresented in which source. So the numbers we are seeing here are the number of data sets that are related to a particular disease term and all of its subtypes in that source. So since we've done this exercise for many different data sources, what we can see here is that IMPORT, which is an immunology database, contains a lot of data related to infections and immune system diseases, as one would expect, whereas the Cancer Genome Atlas, or TCGA, mostly contains neoplasm data. We can also use this sort of way of presenting the data to look at disease classes that are not well studied. So, for example, because we are looking at several different broad disease classes here, we can see that classes like eye diseases or nervous system diseases actually don't have that much public data available in these data repositories. If we compare it to something like neoplasm or cancer data, where a lot of public repositories focus on that type of data. So this is something we can only do if we have used the same vocabulary to tag the same concept across different data sources that otherwise may not have a lot in common. Now, the fourth use case that I'll talk about today is that we are using ontologies to curate not just one type of information and one type of metadata, but we're using ontologies to curate 14 different types of metadata fields which contain different information. And because we're using all of these standardized ways of representing these different metadata fields, we can write very specific queries to find data that is the most relevant for a particular use case. So for example, I'm just doing a callback to the way data are structured in different types of metadata that we have. So I'll do a brief recap. In most of the data that we work with, we've got experimental metadata where we capture information about the experiment. So what was the organism that the experiment was done on, what tissue the samples came from, what was the type of data that was collected, what was the disease being studied, and so on. We also capture metadata about the samples as well as about the molecules or the entities for which the data is being measured. So at the most basic level, because we have done this curation for both the experiment as well as the samples, what we can do is we can write a query that actually filters information across these different metadata fields related to an experiment, such as the type of disease, type of data, type of platform, as well as organism and so on. And we can find data sets that actually satisfy these conditions that have been generated under my particular experimental conditions that I'm interested in. We can go a step further and we can query across not just the experiment metadata, but combine queries across experiment and sample metadata and start asking questions like which data sets study normal versus disease samples in humans through the use of RNA sequencing. So what we are doing here is that we are looking at disease metadata for each sample, but we're also looking at the experiment metadata for organisms as well as data type. And we can use these queries to generate a list of data sets that actually do a normal versus disease sample comparison. We can go even deeper and query across all different types of metadata that we have, the experiments, the samples, as well as the molecular metadata. So here we can begin to ask questions like how the expression of a particular gene changes across diseased and normal samples across many, many different types of data sets. And we can write a query that looks at disease metadata for each sample. It looks at experiment metadata for organisms and data type, as well as measurements of a specific gene to give us a view of what the data contain at a very, very granular level of detail. And all of these levels of querying are actually possible because we have used ontologies to standardize different metadata fields. The last use case that I will talk about today is that ontologies enable us to integrate data and discover new biological connections in the data. So once we've curated different metadata fields like genes, drugs, and diseases, we can then start asking questions about connections between these different metadata fields. So I'll explain this with an example. There are published data sets and studies that look at links between lung adenocarcinoma, which is a subtype of non-small cell lung cancer, and a drug called thapsigargin. And there are independent studies that look at the link between skeletal dysplasia and FGFR3 gene. And because now we're using the same vocabulary for diseases, drugs, and genes across all of the data sets on our platform, we were able to create networks that actually look at relationships between drugs, genes, and disease metadata across thousands of data sets and discover a link between non-small cell lung cancer and skeletal dysplasia, which was not explicitly studied in either of these publications, but was actually embedded within the data itself. And we're now taking this concept further and looking at relationships between a lot more different metadata fields like tissues, genes, cell lines, diseases, and drugs. And we're using these networks to now generate new biological hypotheses. So now one of the things you may have picked on is that we are using pre-existing ontologies for our metadata curation efforts, and that comes with its own set of challenges. So one issue that we run into quite often is that ontologies are not exhaustive. So we often come across terms in data sets that actually do not map to terms present in an existing ontology. And this is something that we have to work around. A second issue is that mappings between different ontologies is incomplete. So for our knowledge graph effort that I just discussed, we have to take on curation of ontologies and build some of these relationships in-house. Now, a third issue is that sometimes ontological concepts don't map very well to biological concepts. What this looks like is that we're using Brenda tissue ontology for curating information about tissues. But this ontology actually contains a mix of terms across tissues, cell lines, and cell types. And it's very difficult to draw a boundary around which term is a tissue and which is not, because it's not clearly defined in an ontology. So if on the application side, we're interested in only tagging tissues and not cell lines, that's it's slightly harder to do. And finally, it's not always straightforward to choose an ontology, since many different ontologies exist for same concepts. And that's something Gabriel also touched on in her presentation. So what we have to do is we have to spend time deciding and choosing the right ontology, and also be prepared to change those choices as we learn more about what our data contains. And this brings me to the end of our presentation. I want to quickly summarize the ideas that we discussed today. We talked about the fact that a lot of data exists in life sciences R&D, but are not usable. And one of the reasons that they're not usable is that metadata are not standardized. And we've seen that we can use ontologies as a key piece of our data pipelines to actually standardize metadata at scale. And we've done this for around 1.5 million data sets. And once you use ontologies to curate and standardize metadata, this can really improve findability of the data by allowing better search and querying experience. And these ontologies then also help us make new biological hypotheses by discovering hidden connections in the data. And finally, using pre-existing ontologies actually comes with its own set of challenges. So data curation teams and ontology experts need to really work together to address these gaps, which is the work that Pistoia Alliance is doing by bringing together different industry experts. And with that, I would like to thank you all for your attention today and look forward to answering any questions.
Thank you very much, Shefali. So we've had loads of questions. Thank you very much for your engagement. Maybe we can start with, we've had a couple on ontology selection, Shefali, relating to your presentation. So why did you decide to use Mesh and Brenda when there are other ontologies that are available? And a similar question about how the ontologies were chosen. So could you give us your thoughts on that?
Yeah, so like I mentioned, that's there's no right answer and there's no right ontology. So when we started curating the data, what we went with is we started with the most exhaustive ontologies, the ones that we thought would contain all of the or as large a selection of terms as we would like to curate, so we wouldn't have to modify them. And then we narrowed from there. So we started with the broadest possible ontologies, and then we modify them for our use cases. So, and we also evaluate and change. So when we started, for example, curating disease, we actually looked and compared between Mesh, disease ontology, DOID, and we so there are two. Sorry, let me refine my answer. So one is we look at how the terms that an ontology contains and whether they are useful, whether they cover the concepts that we are seeing in our data. And the second thing we look at is the connections between different ontologies. So some ontologies are better at defining these links with other ontologies than others. And we try to use the ones that actually define those links a little better.
Yeah, Thank you, Shefali. And maybe taking that a step further, question for you, Gabrielle. What are the key factors for setting up a new one? And
yes, so I mentioned earlier that certainly for Pistoia Alliance point of view, we're not keen on building brand new ontologies and then, you know, going forward, sort of maintaining them, etc. So I think one of the key factors is to find one that already has a good governance model. That's really, really important. You need to know that to ensure the best quality of data in that ontology, that there is a recognized governance model. I think that's very important. And then, as Shefali says, you know, you really want to choose the one, obviously the best fit for the area that you're working in. It needs to be as available as possible, so no kind of restrictions. Yeah, I think really take advice where you can from an ontologist.
Yeah. So we just had a question that's just come in, which actually relates to the answers that have been given. So Daniel Welter has asked, doesn't modifying ontologies for your own purpose make your data less interoperable since you will diverge from other people's?
Right. So when I mentioned that we modify ontologies, we don't actually modify any relationships between them. But the example that I mentioned is with Brenda tissue ontology. So in that ontology, we're using that to tag our tissue terms. So we really filter out cell lines and cell types because we find that other ontologies are actually better. So we are using Brenda tissue ontology, but we're not, for example, using all of the terms within that ontology. We don't actually modify the ontologies themselves.
Okay. All right. And Gabrielle, this relates to your presentation. Is the data model not already provided by CDD Vault?
Yes, I saw that question. I'm not quite sure whether you're referring to the data model that I flashed up in particular, but what I was demonstrating was the type of work that we are achieving in the project team by coming together to produce a data model. Of course, there are many data models. There isn't one answer to any. So I'm sure CDD have a very good data model, but I'm not saying that's right or wrong. I was just demonstrating that this is how the conversation begins. Once we're understanding the relationship between those entities, we can then use that as, I think they like the word scaffold, and get underneath that data model to define use cases for particular domains. So I hope that might have helped with the answer, but if not, let me know.
Yeah. So what I would suggest then, Lucas, if that hasn't answered your question, maybe you could elaborate on that in the Q&A. So Shefali, how do you treat ontologies, or how are ontologies relevant when you're not working with mountains of data? So you're working on small data sets.
Yeah. Yes, I think they are. So ontologies are really important, whether you're working at the level of a single data set or whether you're working with hundreds of thousands or millions of data sets. And a really good example that every biologist would relate to is gene ontology. So even when you're working with one single gene expression data set, you can actually, you don't need to look at the level of individual genes on what's going up or down, but you can actually look at levels of biological processes which are defined in gene ontology to understand biologically what's happening in your data, right? So again, whether you need to use ontologies for every single metadata when working with one data set, not necessarily, but but there are certain types of ontologies that are critical even when you're working with single data set. And as soon as you start getting to multiple data sets, to really keep track of what you have, yes, ontologies are critical. They're not just important when you're working with very, very large numbers of data sets, but even tens or hundreds of data sets.
Yeah, thank you, Shefali. So a question's just come in on the modification of existing ontologies. So do companies typically take, use, and modify existing ontologies? And is it not better to give back useful modifications if they have external applicability? Who wants to start with it? Shefali, yes?
Oh.
Yeah.
Oh. Sorry, I just wanted to say, I completely agree. Yes, it is absolutely. If we're making, so far we've not actually modified ontologies. That's an effort we've just started in our knowledge graph. case study that I just talked about. So, but that's very new. We're still figuring that out, and I absolutely agree that if we are actually making modifications, we would like to actually put them back and submit them for improvement of existing ontologies.
So, Gabrielle, a question for you on seed. What's next for seed?
Okay. Well, what's next this year? As I mentioned, we're looking more broadly across a number of use cases. And in terms of what's next, it's really up to the Pistoia members and the project team to say what are they working on, what do they need next. So very much guided by them. That list that I showed were use cases that the team have prioritised. We can just keep going, to be honest, but it depends very much on the priority of the project team and the Pistoia members.
Okay. Thanks, Gabrielle. And Shefali, would you consider ontologies the most critical piece in adhering to FAIR standards? And what are some other considerations that teams should make?
So ontologies are certainly one of the critical pieces. I wouldn't say that they are the most or the I wouldn't assign a prioritization to that. They're certainly a very key piece. But if we think about FAIR standards as a whole and about data meeting FAIR standards, then we have to think about ontologies in along with all the other aspects like accessibility, like interoperability, for example, and findability. So we have to think about data systems, data structures, how data are stored, along with ontologies in the whole FAIR landscape.
Okay, thank you. So working through these questions, ambiguity, how do you handle ambiguity? For example, different levels of precision on measured value. I've got a feeling that's for you again, Shefali. Do you want to give that a go?
Yes, that's a very challenging problem. Unfortunately, I wish it was easier. It is difficult to actually be. I saw a question somewhere on how do you choose whether, for example, data set should be labeled liver cancer or specific subtype of liver cancer. So sometimes that information is easy to figure out from the information that's available in a data set. So the strategy that we use within our NLP models is we would tag a data set with the most specific term that we are able to assign it to. And sometimes that most specific term is a fairly high level term, like liver disease. In other cases, we're able to be more specific. And when there is when the models are not able to figure it out, then we actually use human experts to kind of resolve that ambiguity as well.
Okay, thank you. Gabrielle, anything to add to that? How do you simplify this? How do we eliminate ambiguity?
No, I can't really add to Shefali's excellent answer.
Yeah, okay. Well, thank you very much. So we're almost at the top of the hour. So what I will do, if I can just seamlessly share my screen. What I'd like to do is to thank Gabrielle, Shefali, and all of our colleagues at Elucidata for their help in putting together this webinar. And I'd also like to thank our attendees for their engagement. And we've had some brilliant questions that have been put in the Q&A function. We haven't been able to get to all of those. Some of them we've answered live through the conversation we've been having, and others we've typed answers to you directly. Those questions we haven't been able to get to, we will follow up with you later. So if any of the topics that we've discussed today have sort of piqued your interest in the Pistoia Alliance, do get involved in our community. We'd love to have your involvement and your feedback. And if you'd like to drop me a line, you can do so using my email address, which I'm sharing with you on the screen. Similarly, if you're not a member of the Pistoia Alliance and this session has generated some interest in joining the Alliance, do do get in touch as well, and we'd be happy to have a conversation with you. So thank you again for taking part in this on this quite lovely summer's day here in the UK, and I hope the weather is good where you are. If there's any follow-up you'd like us to do, either through Elucidata or through the Pistoia Alliance, do get in touch, and we'd be happy to do so. So I'm going to end this session now, and thank you, and I hopefully see you soon at the next webinar. Thank you. Bye-bye.